Good morning, ladies and gentlemen. Thank you all for coming. I would just like to give a quick disclaimer that these sessions are being recorded and will be posted to the United Leukodystrophy Foundation website and YouTube channel. It is a great pleasure for me to introduce our speaker today, Rachel Hickey from Lurie Children's Hospital. There will be a Q&A session at the end, and when that happens, I would just ask to please wait until I bring a microphone to you so that everyone on the Zoom chat can hear your questions too. Thank you.
Hi, everyone. Thank you for that introduction. As she said, my name is Rachel, and I'm one of the genetic counselors at Lurie Children's Hospital of Chicago, and I also am the care coordinator for our leukodystrophy clinic. The title of this presentation is Unlocking Your Family History and Decoding Genetic Inheritance. And so for the next probably 40 minutes, probably not an hour, we will be going through the basics of genetics. We will be covering the inheritance patterns, and then going over how we use that information from the basics of genetics, from the inheritance pattern, to actually decode and interpret genetic testing results. Starting with the basics of genetics, we have to start with a picture that you guys probably remember from your high school biology classes. This is an image of a cell. And within our cells are a bunch of different organelles, which are essentially compartments that have very specific duties, very specific tasks that they need to do in order to keep the cell functioning. The organelle that's most important for us when we're thinking about genetics inheritance patterns is the nucleus, which is in the orange box there. And the nucleus contains our genetic information, which is in the form of DNA. So a gene is a specific sequence that is within the DNA, and we actually can think of that similar to a sentence in an instruction manual. So the DNA sequence is actually just a sentence, and the gene gives us the information on how to make a specific protein that then is used to complete a specific task within our cells, within other cells in the body. And so a normal gene is the typical sequence that we would expect to see in order to be able to make that functional protein. And a mutated gene, or more commonly known as a genetic variant, is a change to that gene sequence that then causes that gene to either not make a protein at all, or to make an abnormal protein, or a protein that's not able to do that specific task. It's also notable that sometimes we can have a genetic variant that does not affect the gene's ability to actually produce a protein. And so not every genetic variant will actually cause any clinical consequences to the ultimate protein product. The DNA is stored into tightly coiled structures that are called chromosomes. And we have a specific genetic test called a karyotype that actually allows us to look at the number and the structure of the chromosomes within our cells. And so if we're looking at this karyotype right here, and if you count those up very quickly, you will see that we have 46 chromosomes. They are organized into pairs, and so you can see one pair here, another pair here. And we have 23 pairs in total. The first 22 pairs are all the same. They are pairs of chromosomes that are organized by size. So chromosome number 1 is the largest, and chromosome number 22 is the smallest. And to talk about the last pair, we have to switch over to this schematic, which you'll see right here is either the XX for females or XY for males. So these chromosomes, that 23rd pair, are the sex chromosomes, whereas chromosomes 1 through 22 are actually referred to as the autosomes. So we differentiate the chromosomes and give them names based on if they're an autosome or a sex chromosome. We have 2 pairs of all of our autosomes, and so therefore we have 2 copies of every single gene that's actually included on those autosome cells. And when we're thinking of the sex chromosomes, so XX for females, so females have 2 copies of every gene on the X chromosome, whereas for males, males only have 1 copy of the genes on the X chromosome, and then they have this Y chromosome right here. So now that we've kind of oriented a little bit to genes and our chromosomes, we can dive into talking a little bit about our inheritance patterns of genetic conditions. If you have ever visited a genetic counselor or anyone that works in genetics, you will know that we love to take pedigrees, which is a visual representation of your family tree. So if we're looking at this pedigree, we can see that there are 4 generations that are listed out. These squares represent the males in the family, and the circles represent the females in the family. And we can use different shading or different nomenclature to help us to track a disease or a symptom that's going through the family. And so really this pedigree allows us to try and identify patterns. The types of inheritance patterns are autosomal dominant, autosomal recessive, X-linked, and then finally mitochondrial, which we'll briefly cover at the end. But one thing to note is that the inheritance patterns are actually named after the chromosomes that those genes are located on. So autosomal means that that gene is located on one of those autosomes. Again, that is chromosomes 1 through 22. And the X-linked are a little more self-explanatory. Those are the genes that are located on the X chromosome. Starting with autosomal dominant genetic conditions are caused by a genetic variant in one copy of the gene pair. So looking at the schematic that's here on the slide, we can see that the individual that is here in pink, this is our affected individual. Looking at their genes right here, they have 2 copies of those genes. And this little orange arrow is pointing to a very small pink mark on one of those genes that actually represents a genetic variant. And so if we follow this in someone's family history, we know that an individual who has an autosomal dominant genetic condition has a 50% chance to pass on their genetic variant to their offspring. And that is dependent on if they pass on the gene that has the genetic change or the gene that doesn't. The other thing about autosomal dominant inheritance patterns is that both males and females can be affected with this condition. An example condition for autosomal dominant in the world of leukodystrophy is LMN1B related leukodystrophy, which is more commonly known as autosomal dominant leukodystrophy or ADLD. And so in that case, we took a very uncreative approach and just named that form of leukodystrophy after the inheritance pattern that we were seeing on that pedigree. Autosomal recessive genetic conditions are caused by a genetic variant in both copies of a gene pair. So again, we're still talking about those autosome chromosomes. And if we look at our little affected individual over here in pink, we can see that she has a genetic change in both copies of this gene. And she inherited these genetic changes from her parents. And so parents of autosomal recessive conditions are called carriers. Carriers just means that you have a genetic change in one copy of your gene pair, but carriers are not affected with these autosomal recessive conditions. And the reason being is that while they do have a genetic change on one of their genes, the other copy does not have a genetic change and therefore is able to compensate for the copy that isn't functioning as well as it should be. So the risks to have an affected child for a couple that are carriers of the same autosomal recessive condition, are 25%. So a 1 in 4 chance that both mom and dad are going to pass on their genetic change to their child. They will also have a 50% chance to have a child that is also a carrier, just like them. So either mom passed on her genetic change or dad did, but the other parent passed on that normal copy. And they will also have a 25% chance to have a child that does not inherit either genetic variant. And so that child is not even a carrier for this condition that's running through the family. One thing that's really important to know when we're thinking about autosomal recessive conditions is that if you have a child that is affected, that does not preclude you from having additional children that are also affected with that condition. So these risks are actually per pregnancy. And so a carrier couple for an autosomal recessive condition may have 5 children and none of them are affected, or may have 5 children and all of them are affected. It really is just dependent on what genetic changes are passed on to their offspring. An example of an autosomal recessive condition is metachromatic leukodystrophy, or MLD. And I chose that as the example condition for autosomal recessive because, as some of you guys may know, we have a new FDA-approved treatment for MLD. And when we're thinking about the individuals that are affected with MLD, who are now having access to treatment, living to adulthood, and then eventually choosing to have children of their own, while MLD is an autosomal recessive condition, this individual who is affected with that autosomal recessive condition will have a different recurrence risk than their parents did. And so this individual's chance to have a child that has the same genetic condition as her is dependent on the carrier status of her partner. And so if she chooses to have a child with an individual who is also a carrier for MLD, their chance to have an affected child is 50%. And that is because this individual will always pass on a genetic change to her offspring. And it is dependent on if her partner passes on a genetic change or if he passes on his normal copy. If their partner is not a carrier, the couple does not have an increased risk to have a child with that condition. That's because their partner has 2 normal working copies, and while mom will always pass on her genetic change, their children only have a chance to be carriers, so all of their children will be carriers for this genetic condition. Moving into our X-linked inheritance patterns, of course, conditions that are X-linked mean that the genes that cause them are on our X chromosomes. And we can subdivide the X-linked inheritance pattern into 2 types, depending on whether or not both of the sexes are at risk to develop symptoms. And so here again is our little karyotype, our little chromosomes, and we can see again that the male has an X chromosome and a Y chromosome, and the female has 2 X chromosomes. I don't know if this is working. Thank you. So starting with X-linked dominant, so an X-linked dominant genetic condition is caused by a genetic change that's, of course, on the X chromosome. But this form of the inheritance pattern can affect both males and females. Truthfully, in clinical practice, we don't necessarily refer to this all the time as X-linked dominant. We typically just call this an X-linked inheritance pattern. But just to differentiate this from the other type of inheritance pattern associated with the X chromosome, for the purposes of this talk, we'll call it X-linked dominant. And so we can see here that, oops, sorry you guys, I am messing up my ability to, I got it, thank you. So we can see here that a female that is affected with an X-linked dominant condition will have a 50% chance to pass on her genetic variant to her offspring, and this is because she has 2 X chromosomes. So she will either pass on the X chromosome that has the genetic change or the X chromosome that doesn't. Both male and female offspring of a female with an X-linked dominant condition are at risk to inherit that condition. However, if this individual, this male, had an X-linked dominant condition, he will pass on his X chromosome with a genetic change to all of his daughters, but none of his sons, because males pass on their Y chromosomes to their male offspring. The other piece with X-linked dominant conditions is that there can be differences between the symptoms observed between males and females. And so most often, males tend to have more significant health concerns, more significant symptoms. And the reason for that is because they only have 1 X chromosome. Whereas for females, we have 2 X chromosomes. And so it acts a little similarly to when we were discussing carrier status for autosomal recessive conditions, where that second copy of the gene may be able to compensate for the copy that isn't working. An example condition for X-linked dominant inheritance patterns is X-linked adrenoleukodystrophy. And for anyone who's been in the field for many years, you may remember that we used to think that XALD only affected males. And we used to call females carriers of XALD. But more research into the field has really shown us that, yes, females are affected with XALD. And instead of being female carriers of this condition, we now call them carrier or females with XALD. Moving to the other X-linked inheritance pattern, which is X-linked recessive, these are genetic conditions caused by genetic variants on the X chromosome, but in this case, only the males are affected, and so females are considered to be healthy carriers of these conditions. A female that is a carrier of a X-linked recessive condition will have a 50% chance to pass on her X chromosome that has a genetic change to her offspring. And if it's inherited by a male offspring, he will be affected. But if it is inherited to a female, she will also be a healthy carrier for that condition. A good example for X-linked recessive is Pelizaeus-Merzbacher disease, and this is a condition where we very often find that mothers of children who have PMD are carriers and really never knew that they were carriers because they never experienced any symptoms themselves. Okay, and moving into the final inheritance pattern that I wanted to talk about today, we have to go back to our beautiful picture of our cells. And if you remember, we talked a lot about how the nucleus contains our DNA, but it's not the only organelle within our cell that has DNA. There also is DNA that is located in the mitochondria. Again, high school biology, something everyone tells me is that they remember that the mitochondria is the powerhouse of the cell. It also has its own DNA. The karyotype, or the autosomes in the sex chromosomes, are located in the nucleus of the cell. The mitochondria has its totally separate genome. And the one thing about mitochondrial inheritance is that we get our mitochondrial from our mothers. And so a mother that has a mitochondrial disorder will pass on her mitochondria to all of her children. Mitochondrial disorders and classic leukodystrophies are very separate. They have separate pathophysiologies. But one thing that I've seen get a little complicated is that some of the classic leukodystrophies have effects in the mitochondria, and some of them even have the word mitochondria in their name. But it's important to note that those conditions actually don't have a mitochondrial inheritance pattern, even though they affect the mitochondria in some way. And a good example for that is the mitochondrial tRNase synthetase deficiencies. So those are actually genes that are located on our autosomes that are associated with autosomal recessive inheritance patterns. And so it's really important to know the exact inheritance pattern of the condition that's running in your family, because it can really help to stratify the risk of who is actually a potential to be affected with this condition. And now that we've talked a lot about inheritance, we have to go into a discussion of, are genetic variants always inherited? And the answer to that is no. You may have heard that all humans are unique. And the reason that we're all unique is because we all have unique genetic changes in comparison to one another. Thank you. These genetic changes are referred to as de novo. De novo is a Latin phrase meaning in the beginning or anew. And de novo genetic variants are a new genetic variant that occurs in the child that was not inherited from either parent. So you can see that neither of these parents have a genetic change in either of their gene pairs, but this little kiddo does. These genetic changes, these de novo changes, typically occur at conception. And I mentioned earlier that, these de novo changes are what make everyone unique, and we all have de novo changes in comparison to our parents. It just matters if that de novo change is in a gene that's important for health, and if that variant in that gene actually affects that gene's ability to create a protein. An example of a de novo genetic condition is an infantile form of Alexander disease, where most often we see that there's a child affected and neither of their parents are affected. And for de novo genetic variants, we quote that there is a recurrence risk of 1%. So certainly not as high as the recurrence risk that we've seen for autosomal dominant conditions, autosomal recessive. But we can never quote 0% risk. And the reason why we cannot quote 0% risk, can you switch it to the next slide, is because of a phenomenon called germline mosaicism. So mosaicism refers to an individual having 2 or more cell lines within their body that all have different genetic content or different genetic changes. And so if we're looking at this cells on the left, you can see that all of them are dark purple. So all of them have the same genetic content. But looking over here at this other cells, we can see that some of them are dark purple, but some of them are light purple, which is used to indicate that these cells have differences or different genetic changes than their counterparts. We named this mosaicism because it reminded somebody of a mosaic tile. So if you've seen any of those, there's a bunch of different tiles that are all put together in order to make one mural, essentially. And so germline mosaicism refers to having genetic variants in only some of the germline cells. So those are the egg and the sperm. And we really are not able to ever rule out the possibility of germline mosaicism. It is very rare, but that's why we're never able to quote you as less than 1% if you have a child that has a de novo genetic change. So now that we've talked about the basics of genetics and different types of inheritance patterns, how do we use that information to then apply it to genetic testing results in our patients? And we have to kind of start a little back in talking about how we classify genetic changes. So as I mentioned earlier, not every single genetic change that can happen in your genes is actually contributing to your health. The classification scheme for genetic variants is very complicated, but simplistically we can break it down into 3 possible types of classifications. The first is pathogenic, and so a pathogenic genetic change means that this genetic variant is harmful to that gene. It is causing that gene to not make the protein that it is supposed to make, and then that protein is not able to do the task that it is supposed to do. The opposite of a pathogenic is essentially a benign genetic change, and this is a type of genetic change that does not affect the gene's function in any way. So that gene with a benign change is still able to make a protein product. And in between pathogenic and benign is this gray zone over here called a variant of uncertain significance. And a VUS is a genetic change that we can see on genetic testing. We can find that you have a variant in this gene, but we just don't know if that variant is actually causing that gene to not make its protein product. Most often, this is because we just don't have enough data on that variant, or we've just never seen that variant before, and so we don't know how it relates to human disease. One way that we can kind of get a better understanding of a variant of uncertain significance is by actually testing other family members to help see if that variant is tracking with disease in this family. And so case 1, we have a small pedigree here. Patient 1 is shaded to indicate that they have some symptoms, something that we're suspicious might be genetic. And so as an example, patient number 1 has a suspected leukodystrophy on MRI. And we do genetic testing for the proband, which is patient number 1, which reveals a variant of uncertain significance in the GFAP gene. And we know that pathogenic variants or disease-causing variants in this gene are associated with Alexander disease, which was the example that we used for our de novo genetic variants. And so in a child with Alexander disease, infantile Alexander disease, what I would expect to see in their pedigree is that neither of these parents have that same genetic change. And so we can do parental testing, and if that genetic change is found in one of the parents, in this example it's found in the mother, that probably gives me more assurance to say that this VUS is probably not causing any health concerns in this family tree. If this mother doesn't have any signs or symptoms of Alexander disease, then this VUS is most likely benign. In the case scenario where we actually find that this genetic change is not present in either parent, then we are able to say this VUS is de novo in this patient. And so that is more in line with the inheritance pattern that I would expect to see for an infantile Alexander disease. And so the parental testing in this case have given us the ability to say this is a possible diagnosis of Alexander disease. But you'll see that I left a lot of little asterisks here, and that is because we need to be very careful when we are assigning a disease status to someone based off of a variant of uncertain significance. And in a lot of ways, we need to rely on the patient's clinical history in order to help us to really say whether or not this VUS is causative, because there's still a possibility that this could be benign, even if it is looking like it's matching up to that expected inheritance pattern that we're looking for. I will get back to that double asterisk, but moving into case 2, again, we have a little pedigree here with patient number 2 who is symptomatic. And patient number 2 also has a suspected leukodystrophy on their MRI. And so genetic testing for proband or patient 2 reveals that they have a pathogenic variant, disease-causing variant in a gene called ARSA. And they also have a VUS in the same gene. And pathogenic variants in ARSA are associated with metachromatic leukodystrophy, which again is the example that I had used for the autosomal recessive inheritance pattern. So simplistically, what I would expect to see in somebody who has MLD is 2 genetic changes within that ARSA gene. But going back to our case where we have only done genetic testing for this child, whenever we see that there are 2 genetic changes in this patient, we don't have enough information to know if those genetic changes were either inherited from only one parent or one was inherited from mom and the other was inherited from dad. So we need to do further family testing in order to know how those variants are actually tracking within this family pedigree. So on this slide, we have a chromosome pair or a gene pair right here. And for patient 2, we know that she has 2 genetic changes in ARSA. It is possible that both of those genetic changes are on the gene that she inherited from her father. So these represent little genetic changes, and if this is the case where 2 variants are on the same gene pair, we call that in-cis. Whereas if there is a genetic change that came from the father and a genetic change that come from the mother, we call that in-trans. And so for autosomal recessive conditions, what we're looking for is that those genetic variants are in-trans, so those 2 variants are inherited from either parent. And so we can do parental testing, and if we do find that scenario where both of the genetic changes are inherited from one parent, that is not suggestive of an autosomal recessive inheritance pattern. But if we find that one of the variants is inherited from the father and one is inherited from the mother, this gives us a little more information, a little more evidence that this inheritance pattern is suggestive of the autosomal recessive condition that we're looking for. And so we now have a possible diagnosis of metachromatic leukodystrophy in patient 2. However, we have these little asterisks here because we still need the clinical information for our patient in order to better understand if this VUS is really something that's causative. Genetic testing is very imperfect, and it's really because we don't know every single type of genetic change that can potentially contribute to human health. And so in some cases, we rely on clinical findings to help us to make a diagnosis. And so going back to case 1, patient 1, who had possible Alexander disease, some of you may know that Alexander disease actually has a very specific area of the brain that it affects. And so in that patient who had a VUS, a de novo VUS, and also MRI findings that are very suggestive of Alexander disease, that helps us to give enough evidence to say, this is in the inheritance pattern we expect. We also are seeing the clinical findings that we expect for Alexander disease, and so that's most likely a true diagnosis. For patient 2 with metachromatic leukodystrophy, While there are some specific MRI findings that we can see with metachromatic leukodystrophy, this condition actually has very specific biochemical findings. So they have specific enzyme levels and specific substrates that we're actually able to measure to give someone a biochemical diagnosis of MLD. And so for patient 2 who has a pathogenic and a VUS that are in trans, so in the inheritance pattern that we're looking for, and biochemical testing that is suggestive of that diagnosis, that's usually enough for us to say this is a true diagnosis of MLD. However, if that individual had normal biochemical testing, even though they have genetic changes that are in trans, if they don't have the suggestive biochemical testing, then it's more likely that that VUS is not affecting that individual, and we probably need to do further genetic testing in order to get to the bottom of their symptoms. And so for the final case that I wanted to talk about today, I wanted to flip the script of going not from someone already having genetic testing results, but for someone coming to clinic who has MRI findings that are suggestive of a specific clinical diagnosis. And so patient number 3 has MRI findings that are suggestive of Pelizaeus-Merzbacher disease. And the question that we have is patient 3's younger sister at risk to potentially be affected? Pelizaeus-Merzbacher, if you guys remember, is the example that we used for the X-linked recessive inheritance pattern, where only the males that inherited a genetic change on their X chromosome are affected, and the females that have that genetic change are healthy carriers. And so for our pedigree, if this truly is Pelizaeus-Merzbacher disease, while patient 3's younger sister may be at risk to be a carrier, she would not be at risk to be affected. But one of the things that's complicated in medicine is that a lot of genetic conditions can look very similar to each other based on their clinical presentation. And so while patient 3 has MRI findings that are suggestive of Pelizaeus-Merzbacher disease, there is a genetic condition that also can present in the exact same way. And we have decided to be as confusing as possible and name it Pelizaeus-Merzbacher-like disease. So both of these conditions can present very similarly, but importantly, they have very different inheritance patterns. So Pelizaeus-Merzbacher is caused by genetic changes in PLP1 and is X-linked recessive, but Pelizaeus-Merzbacher-like disease is actually caused by genetic changes in GJC2. And GJC2 is located on chromosome 1, which is one of our autosomes. And so this condition is autosomal recessive. And so if we go back to case 3, the question is patient 3's sister at risk to be affected? And the answer to that is that we probably need to figure out the genetic etiology of patient 3's MRI findings in order to really know who else we need to screen within this family. And so, in summary, genetics is very complicated. Sometimes it is very straightforward, sometimes it is not. You guys may know that a lot of the current management guidelines for different leukodystrophies all say that an individual with this condition will need consultation with a genetic counselor or genetic specialist. And the reason for that is that we are here to help to clarify the significance of your genetic testing results and also to help you to understand what is the recurrence risk for other members of your family. And that really brings us to the end of my presentation. I would certainly be happy to take any questions from the audience, or you can always find me afterwards and ask me a question then if you would like. But thank you.
At what point does the scientific community say, okay, this, and then the individual does turn out to have symptoms? Mm-hmm. At what point does that variation come to be called?
Yeah. So the question is, if you have somebody who has a VUS, and we ultimately determine that the patient does truly have that genetic condition, at what point do we upgrade that VUS to disease-causing? And that is a hard process. A lot of it is actually individual clinics reporting back to the genetic testing laboratories that this genetic change that you call the VUS, we actually feel like it causes disease in this patient. Another thing that we do is that individual clinics or big medical centers will often publish their cases, and so publication of a VUS in the literature in association with genetic disease is also a way for our laboratories to actually upgrade that classification from uncertain to truly pathogenic.
We do have a question from the chat, and I'll do my best here. Never knew I had UBTF leukodystrophy. How can we advocate for early testing? I live in North Carolina, but my son was born in Virginia. I am autosomal dominant, and my pattern is mosaicism and VUS. We never knew I had this till I had my son, and he found out at 17 months that he was full mutation of UBTF. No one suggested genetic testing because we had no history. How is it possible that we, my son and I, are the only ones with an inherited mutation? All other variations of a mutation are de novo?
Yeah. First, that's a great question, and thank you to that audience member for sharing that story. As I said, in genetics, it is very complicated, and not every family's case will fit very neatly into those inheritance patterns that I shared. I think the other part of the question was, how do I advocate for myself and how do I ensure that there are more testing for cases like mine? And really the answer is to do exactly what that individual is doing, which is by speaking out and being an advocate for yourself and sharing your story. That's really the only way that we will continue to learn, both in the medical field and also just about other individuals who are affected with these more rare forms of these disorders. Okay, thank you guys.